translation. So we have seen what is replication, transcription. Now coming to translation. So in case of replication, DNA is synthesized from a DNA. So that is replication. Coming to transcription, RNA is synthesized from DNA. We call it as transcription. And that RNA, when it acts as a template for the synthesis of protein, we call that process as translation. So synthesis of protein directed by mRNA template is called as translation. So simply you can say that synthesis of proteins, and that process we call it as translation. Okay. Coming to, so here we can see that, so both here we require mRNA, rRNA, tRNA for the synthesis of proteins. So in addition to that, we require amino acids. So these are the important components required for the synthesis of proteins. Coming to, so before going to details of translation, first we'll discuss about what is genetic code. Right? So as I told you, mRNA template directs the synthesis of protein. So protein can be synthesized from the sequence which is there in the mRNA. The sequence of nucleotides in the mRNA, we call it as codons. So codon is a sequence of 3 nucleotides in an mRNA is called as codon, and those codons in an mRNA, they specify for a particular amino acid. So this is called as genetic code. So sequence of nucleotides in an mRNA which specifies a particular amino acid so that a protein or polypeptide can be synthesized. So those sequence of nucleotides, we call them as genetic code. So basically sequence of 3 nucleotides, we call it as codon. So each mRNA has many number of codons. Each codon specifies a particular amino acid. Right? So that is genetic code. And genetic code, so it is a, as I told you, it is a triplet codon, sequence of 3 nucleotides. So here the first base, in case of when you are writing the codons, triplet codons, the first base we have to take at the 5' end, it is U, and second base will be UCAG, and the third base will be, you have UCAG. So when you consider the first amino acid with a triplet codon, that is phenylalanine, so the codon for that phenylalanine is UUU. So that codes for the phenylalanine. Right? So first base in case of phenylalanine is U, and the second one is U, and the third one is U. So when you take the stop codons UAA, UAG, UGA, so they also have 3 codons, 3 nucleotides. So UAA, the first base in case of stop codon UAA is U, and the second base is A, and the third base is A. So this is how sequence of 3 nucleotides will code for a particular amino acid. So each codon codes for only one amino acid. So whereas, for example, if you take UUU, it codes only phenylalanine. It won't code for any other amino acids, isoleucine, tyrosine, tryptophan. So like that, it won't code for any amino acid. So that is the significance of genetic code. So coming to significance, it is universal. When you take this genetic code and sequence of codons, so each codon codes for a particular amino acid, where in any animal the same sequence codes for the same amino acid. Right? So that's why we call it as universal. When you take unambiguous, so what do you mean by unambiguous? Here, so each codon, as I told you, codes only one amino acid. So that feature of genetic code is called as unambiguous. Coming to degenerate. So genetic code is degenerate. So what do you mean by that? Degenerate means, so as I told you, so each codon codes for a specific amino acid. One amino acid can be coded by different codons. So that feature of genetic code is called as degenerate. So genetic code is degenerate. That means many codons, they can code for a single amino acid. And the other important feature of genetic code is it is non-overlapping and commonness. So there is no commas, punctuations when you are reading the genetic code. So they will start from 5' end and it ends in the 3' end. So it is a continuous sequence. So there is no punctuations and commas in case of genetic code, and it is non-overlapping. So that's these are the important features of genetic code, right? Coming to translation, right? So in order to synthesize a particular protein, the mRNA for the synthesis of each protein, we require a particular amino acid. So when you take, for example, glucokinase, for the synthesis of glucokinase, we require mRNA which has sequences which code for the amino acids in that glucokinase, right? So when you take hexokinase has a separate mRNA with different codons. So each protein has a particular codon. It has a message in the mRNA. That message is translated into corresponding proteins, right? Coming to translation process, the important steps in the translation are: first you have to activate the amino acid, right? That is the first step required in the translation. After that, initiation, elongation, termination, and then we'll get a product, nascent polypeptide chain, nascent protein. So that is not biologically active. We have to convert that into biologically active form by a process called as post-translational modifications. So these are the important steps in case of protein synthesis. So what are those steps? Activation of amino acids, initiation, elongation, termination, and post-translational modifications. So we'll see in detail about each process. Coming to activation of amino acids. So as I told you, for the synthesis of a protein is nothing but polymer of amino acids. So it is made up of amino acids, right? For the synthesis of protein, we have to polymerize many amino acids. We have to link many amino acids. So they have a particular sequence, and that sequence is determined by the sequence in the mRNA, right? So the sequence in a protein is determined by the sequence in the mRNA. So those codons in the mRNA will specify the sequence of amino acids in a given protein, right? But in order to insert those amino acids in the protein synthesis, first we have to activate those amino acids, right? So activation of amino acids is very simple. Just in this activation of amino acid reacts with tRNA, and this process requires ATP, and that will be converted into amino acyl tRNA. In case of eukaryotes, the tRNA which is synthesized initially, it is methionine tRNA. The first amino acid that is incorporated in case of eukaryotes is methionine tRNA, and the codon for the methionine tRNA is AUG. So that methionine reacts with tRNA, and that requires ATP, and this is catalyzed by the enzyme called as amino acyl tRNA synthetase. So that amino acyl tRNA synthetase converts methionine plus tRNA plus ATP into methionine tRNA. So that methionine tRNA, we call it as the active form of methionine. So that's how we are activating methionine, by converting that into methionine tRNA. Right? So that is first happens in case of translation. We have to activate each amino acid. Just simply you will add that amino acid to the tRNA, then it gets activated. And coming to initiation of translation. So what happens in case of initiation? So first we have activated the amino acid. After that, in initiation, the first step of initiation is we have dissociation of ATS ribosome. So that ATS ribosome dissociates into two subunits, one 60S subunit and the other one is 40S subunit. So that dissociation is required for the initiation of translation. Right. So what happens after the dissociation? So 40S and 60S are dissociated from the ATS subunit, ATS ribosome. Then to prevent the reassociation of these 40S and 60S subunits, two initiation factors, they are bound with the 40S subunit. Those are initiation factor 1 and initiation factor 3A. These two factors will bind with the 40S subunit, and the significance of these factors is they prevent the rebinding, reassociation of 40s and 60s subunits, right? So after that, what happens to the 40s subunit? This 40s subunit then reacts with a ternary complex. So that ternary complex has three components. Those are initiation factor 2 and GTP, and the other one is the activated amino acid, that is methionine tRNA. So that methionine tRNA, initiation factor 2 and GTP, these three form a ternary complex. This ternary complex, this binds with the 40s subunit, and it results in the formation of a 43s pre-initiation complex. So that 43s pre-initiation complex now reacts with to this 43s pre-initiation complex, mRNA binds, and when mRNA binds, it is converted into 48s initiation complex. So 43s initiation complex is converted to 48s initiation complex, and this conversion is brought when mRNA binds with the 43s initiation complex. So this binding of mRNA to the 43s pre-initiation complex is favored by 5' cap of the mRNA. So that 5' cap of mRNA helps in the facilitates the binding of mRNA to that 43s pre-initiation complex, so that it results in the formation of 48s initiation complex. Now what happens to 48s initiation complex? So this 48s initiation complex, it scans the mRNA and searches for the initiation codon AUG, right? So there it searches for the AUG codon and binds with the AUG codon. Now after that, that 48s initiation complex again reassociates with the 60s subunit, and that is converted into 80s initiation complex. Right? So initially, when you are starting initiation, 80s ribosome is dissociated into 60s and 40s. So that 60s subunit, it reassociates with the 48s initiation complex, and it forms the 80s initiation complex. So this is what happens in case of initiation of translation. So there is a dissociation of ribosomes into 40s and 60s, and there is formation of 43s pre-initiation complex, 48s initiation complex, and finally 48s initiation complex again reassociates with the 60s subunit and is converted into 80s initiation complex. For the conversion of 48s initiation complex into 80s initiation complex, we require initiation factor 2, GTP, and initiation factor 5, so which has a GTPase activity. So that GTPase activity helps in the reassociation of these two subunits, right? So here energy is utilized for the association of this 40s and 48s and 60s subunits to form 80s initiation complex. So this is what actually happens in case of initiation of translation. Now we'll see what happens after initiation. After initiation means, so at the end of initiation there is formation of 80s initiation complex. That initiation complex has, say, RNA at the P site. So 80s complex has two sites, P site and A site. So P site is called as peptidyl site and A site is called as amino acid site. So that A site accepts the incoming amino acid tRNAs, and P site has the peptidyl tRNA. Now that P site, after the formation of 80s complex, now it has methionine at the methionine tRNA is at P site. Right? Now coming to elongation, so this 80s initiation complex with methionine tRNA at the P site means A site is free. So that A site is now occupied by the new amino acyl tRNA. That amino acyl tRNA comes and occupies the A site of the 80s initiation complex, and so this is what happens in case the first step of the elongation, right? After that, what happens? The amino acid in the P site and the amino acid in the A site, they are joined by a peptide bond. So amino acid in the P site forms a bond with the amino acid in the A site, so that now the tRNA in the P site is free because the amino acid formed a bond with the peptide bond with the amino acid in the A site, right? And this bond formation is catalyzed by an enzyme called as peptidyl transferase. So 28S ribosome, 28S subunit of 60S ribosome has this enzyme activity. So that catalyzes the formation of the peptide bond between the between the amino acid in the P site and the amino acid at the A site. Right. Now, after the formation of peptide bond between the amino acid at the P site and the amino acid at the A site, now the tRNA in the P site will be removed from the P site, and ribosome translocates to the next codon, so that now P site is occupied by the peptidyl tRNA. Again, one more amino acid tRNA comes and occupies the A site. Right. Again, the peptidyl tRNA in the P site forms a peptide bond with the amino acid in the A site. Again, it translocates. Again, it goes to the next codon, where that codon specifies a particular amino acid, and that tRNA comes and binds, occupies the A site. So this is how translocation, elongation in the translation occurs by the sequential addition of the amino acids, so based on the codon present in that mRNA. Right? So this is what happens in case of elongation. So in case of elongation, for the proper positioning of tRNA in the P site, we require elongation factor 1, and there is one more elongation factor, that is elongation factor 2. So that helps in the peptide bond formation, and also the other function of this elongation factor 2 is that removes the tRNA at the P site. So for the removal of tRNA at the P site after the formation of peptide bond, right? So that is the function of these two elongation factors 1 and 2. So this is the whole mechanism that occurs in the elongation. Right? And coming to termination. So after elongation, so during elongation, each codon specifies a particular amino acid, and that occupies the A site. After that, the amino acid in the amino acid tRNA, peptide tRNA at the P site forms a peptide bond with the amino acid in the A site. So this sequence of reactions occurs until it comes in contact with a codon called as stop codon. So there are 3 stop codons: UAA, UAG, UGA. So these are called as stop codons. So whenever a stop codon occupies the A site of the ATS complex, then termination occurs. So whether it may be UAA, UGA or UAG. So whatever in stop codon it may be, but whenever it occupies the A site, then it leads to termination of translation, right? Protein synthesis, termination of protein synthesis occurs. So these are the this is about the termination of translation. Now coming to post-translation modification. So after termination, what happens? The releasing factors will release the peptides, polypeptide or protein from the ATS complex. So after that, tRNA is released, mRNA is released, rRNA is free, and all the initiation factors are released from that ATS complex. Right? So that polypeptide or protein which is synthesized at the end of termination, we call it as nascent polypeptide, or it is functionally inactive. We have to modify that protein or polypeptide so that it becomes biologically active protein. Right? For that, we have to do some modifications. That protein undergoes some modifications. So those modifications are called as post-translational modifications. So after the synthesis of protein, that protein is undergoing some modifications. So that we call it as post-translational modifications. Right. So what are those post-translational modifications? So there are some important post-translational modifications. Those are proteolytic cleavage. So the unwanted sequences in the protein, they are removed by proteolysis. So proteolytic cleavage, gamma carboxylation. So when you see clotting factors, they undergo a process called as gamma carboxylation. Actually, they are inactive form in blood. So whenever they are required, they are activated by post-translational modification called as gamma carboxylation. So those clotting factors, for example, when you take clotting factor 7, clotting factor 9, 10. So those clotting factors, they are activated by gamma carboxylation. So it's nothing but a post-translational modification. And hydroxylation, phosphorylation. Hydroxylation. So when you see collagen is activated. It undergoes post-translation modification, and the most important reaction modification in case of collagen is hydroxylation of proline and lysine. So that hydroxylation reaction converts the immature collagen into mature collagen. Right? So actually it is a synthesized as pre-pro-collagen, and after removal of the signal sequence it is converted into pro-collagen, and finally that will be converted into collagen. So pre-pro-collagen, pro-collagen, collagen. So collagen is the active form of protein, that is pre-pro-collagen. Right. So that is hydroxylation. Coming to phosphorylation, so the activity of enzyme may be increased or decreased by adding a phosphate group, that we call it as phosphorylation. And glycosylation, the addition of some carbohydrate moieties will be taking place in case of glycosylation. So these are the important post-translational modifications in case of translation. So inhibitors of protein synthesis. This protein synthesis is inhibited by some important agents like tetracycline, puromycin, erythromycin, and streptomycin, diphtheria toxin, chloramphenicol. So these are the important agents which inhibit the translation protein synthesis. So most of them, they are used as antibiotics. Right. So these are the inhibitors of protein synthesis translation. Right. So till now we discussed about protein synthesis, what is genetic code, what are the phases of protein synthesis, how initiation occurs, how elongation occurs, and why the termination of translation occurs, and what happens to a polypeptide which is synthesized after the termination, how it undergoes some modifications, how it is converted into the active form of protein, right? And what are the agents which inhibit the protein synthesis.